Thanks a lot. It's very nice being here. Last time was 2 years ago, and I remember this is really a highlight. Very nice conference. Thanks, Jim. Okay, so today I will talk about the visual predictions, and to start, let's talk about the kind of mystery. How does the brain get knowledge about the world? How does world knowledge is represented in the brain? Once you look into the brain, you see, of course, lots of connections, and only 5% is actually sensory input. So most of the time, the brain is sending and receiving messages between areas. It's one of the mysteries to understand what these message passing is, and we don't really have so many good experiments to do that. So one aspect we haven't talked too much about is this kind of world knowledge, the different images, how they're represented and maybe labeled in a feed-forward network. That was something we have seen yesterday. But how does the brain, how is it even motivated to have a grasp, an understanding, knowledge about the world? And predictive coding framework applies a very interesting way where you get this for free in a way. And the idea here is that any organism creates internal models, and the internal models need to represent in a meaningful way the outside world. And they do this by message passing in a hierarchical, potentially less hierarchical, different ways. But in some way, it will need to represent some useful aspects of the outside world for the organism. And the currency to see whether an internal model is a good internal model is the prediction error. And if a system is able to create internal models that are actually able to predict the outside, the incoming sensory input, and in this sense, like slow feature extraction, knowing that this ball is moving faster and this is a person, and if you grasp the punchline of a joke, and if you understand the escalators are usually moving and so on, you have essential world knowledge in the system. The system has somehow managed to make these predictions and will appear as very intelligent, even if it's using all kinds of tricks, but essentially minimizing the prediction error in forward generative models is the currency that it needs to fit. And by doing this, you certainly have an intelligent system. But is the brain actually using this? So therefore, I'll come and use a few experimental procedures to use brain imaging to see whether indeed we find evidence that the internal models are generating models that are presenting all the way down the hierarchy to the entrance level. So that would be the question. And as I said, for perception, the system knows it has a useful model about the world if it reduces the prediction error. But then, more meaningful, it wants to create an prediction over the near future, possibly the far future, and the further it can run predictions, the more intelligent it will be and the more flexible to live in challenging environments. And again, the models need to predict a changing environment, so just grasping an image will not be enough, but being able to understand the kind of transitions in near future is the essential world knowledge. And one way of doing this, while the system is developing, is to challenge itself like 4 times a second by bringing about changes in the world. So the changes in the world aren't fast enough to acquire this very good understanding. And so therefore, we make lots of saccades, and the saccades change everything. And only if we have a very good understanding of the world, the things change in a predictive way. So therefore, we're using an experiment in which we do these changes and see whether V1 is informed of the predicted changes and processes prediction error to this. So that's the outline for the talk. So let's start with the first simple example. So we presented 6 images for 100 milliseconds, and they're from a movie, so they're in a natural sequence presented here. And so this goes through the hierarchy of visual areas. It's a complex stimulus, so it's not only not only contours, has complex features and so on, different moving objects. So the system will be at work and have an understanding of which features change faster and slower and so forth. But will it also use this to make a forward prediction? And so we tested this, a movie, then we introduced a small break of 400 milliseconds, and then a test stimulus that was taken from the movie after 400 milliseconds. And we tested, so that's an example. You have a small, you could say like an eye blink, or if you're in the cinema, that's something that's often done in commercials. You have a clip for 1 second, then it flips to another clip for 1 second, and so on. And it keeps you on the toes. There's always this prediction error. So you know it's enough to understand the gist. It's a James Bond, and here it's a romantic movie, and then there's a sport movie, and so on. And you understand the agents, you recognize the people, you make forward predictions, and then it jumps to the next one. So this is a kind of a testing whether we have forward projections over this 400 milliseconds gap. And to test this against a condition in which we took an image taken from the same movie, same gist, but from a different time point. So I made it a little bit more visible here. So it's 4 seconds later. So there was the fMRI experiment, but since it's not very much controlled, these two last images, we jittered the prime, so to say, and now it's not really a continuous movie, and so therefore the predictions are not very easy it all has the same gist, but this one is not more predicted than this image after a sequence in which the images are jittered. So we used 24 different scenes here, a few examples from the market scene. The one I showed you before was a bit different timing, so these are the real timings. You need to get used to it in the experiments. So we had 400 milliseconds. We also had an experiment of 800 millisecond gaps. And again, you see these jittered non-expectancy conditions. And so what we can see is that early visual cortex in 10 subjects, you find for the expectancy match condition, slight attenuation in the activity. So that's what we were looking for. You also see this in V3 ventral. And those are the ones you see in retinotopic areas, and we also looked whole brain analysis. And in whole brain analysis, you see the medial parietal, the precuneus and medial temporal lobe, and also around MT, where the forward models are generated, and especially the combination of expectancy match is giving the lowest signal. So this is a control experiment, a variation. We had 800 milliseconds, and now a more parametric variation of the test stimulus. We used one that's 300 milliseconds, so early from the gap. If this adaptation would just be related to similarity, then this one should be the most adapted stimulus. If it's actually a precise forward prediction, then the 800 milliseconds should be the most adapted, and as a control, we have again a 4 second later from the movie. And what we find is in V1, even in V1, for these complex visual stimuli, we have the highest adaptation for the matching condition of 800 milliseconds. And this resembles some of the findings of Dave Parrott, who has recorded an IT cortex in monkey and shown these kind of grasping movements to the monkeys in the natural sequence, which is shown in the dotted line. And if those images are presented as single images, separated from each other, you see each responses in these solid lines. And so if they come in a natural sequence, the activity quickly adapts. And another way of showing this is the response to the last stimulus. So now here's the response shown to the last stimulus, which is a forward oriented phase. And if this phase is at the end of the natural sequence of a rotating head, then it creates the least activity. If it's following a jittered sequence, it is not predicted. or if it's following numerical sequences, as an example. So this is an example for showing that we have something like a simulation in real time that is ongoing during perception and making forward predictions for roughly a second in the early visual cortex. And now I will move to the second example that I said, in which we do action and see whether the generative models are anticipating the changes that come about in the world by the planned actions of eye movement. And to do this, I will start with a apparent motion illusion and highlight the study from Dartmouth. Won-Mok Shim did this beautiful study in which I'm not sure if this is a real resemblance of the movie, but I just tried to make it from the paper. So this is kind of an apparent motion with the grating, and what they found is that you find in the apparent motion trace activity that is that can be decoded, that a new feature is created along the apparent motion path, and the new feature is the matching vertical, the intermediate vertical grating, if you like. So the generative model is not making an adaptation of the neighbor features, but making a useful interpolation between those features of orientation to say, you know, for this illusion, I'm expecting that there's a continuous change. And there are various controls, and it follows a gist of experiments that we have been performing over the last 10 years or so, and it's a very beautiful design. So what we're gonna do is a very simple apparent motion of a dot that's moving up and down, like here, but the subjects are asked to fixate on the red cross that you can see first on the right, and after a while it switches to the left, and then you need to make a saccade to the left. And what we did is that just right after the saccade is executed, we make the experiment such that there's only one more image shown, and let me highlight this. I think, yeah. So here again, on the right, now saccade to the left. As Patrick Kavanagh has shown, you'll continue to have very nice continuous predictive apparent motion perception. It sometimes tilts a little bit, but otherwise it stays. But what we have also shown is that our system creates predictions along the apparent motion trace, and to test them, we apply a test stimulus in the right timing or slightly off timing along the apparent motion trace. And this is our test stimulus. So it's in between. We only show this after the saccade and then the final stimulus. And then we ask the subjects, did you see the target or not? So one more time. On the right, apparent motion, saccade. Was there a test stimulus or not? Did you see a test stimulus? Yeah? Okay. I tweeted the timing is not quite like in the experiment, but almost. Okay, so it's a very stable finding. We have done this now in various ways, and this is highlighting a study that was done in collaboration with Philipp Sterzer in Berlin, and they tried it with schizophrenic subjects. And it shows that if you have the test stimulus in the right timing, this is a space-time plot, so the dark ones are the right timing, then control subjects, shown here during apparent motion, as much as schizophrenic patients, show an advantage in recognizing the one that's presented in the right timing. And the one that's off, showing at the wrong timing, is not as well detected. So that's a stable finding. And we have done fMRI in 2010, where we showed that the one that is recognized, so the in-time stimulus, produces less fMRI signal in area V1. So that's inconsistent with the idea that the more activity you have, the better you perceive it, but it's consistent with the predictive coding framework that if something is predicted, it can be processed more efficiently, still be detected, but it's just confirming your prediction. If it's slightly off in timing, it's a surprising stimulus, and it depends on the kind of avalanches that follow, whether this will be detected or whether it will be regarded as noisy stimulus. So in this case, it's not as well detected, even though it's created an error signal. So now we use this in our combined experiment with the saccade. We map out the region of the target stimulus, so that's shown in blue. It's adjacent to the lower apparent motion stimulus. So now we are interested to map out fMRI signal only at this position and only after the saccade. So the whole creation of the internal model is done in the other hemisphere. As I showed, the fixation cross was on the right, so the apparent motion stimulus is on the left. So in the right hemisphere, you're processing the internal model of apparent motion. You process the prediction, and you send the prediction down the visual hierarchy to primary visual cortex. Now you plan a saccade. Do you also transfer your prediction of this internal model to the new hemisphere in time for the next stimulus to arrive, to then be processed in a different way? So if you do that, that's really efficient coding. It would be predictive. It would transfer that to the other hemisphere. And yes, what we find is the following. At this location, the enhancement to activity during apparent motion is the difference to the baseline. This is in conditions where we're not showing the test stimulus, the target stimulus. You still have an enhancement of activity, and that is related to the internal model. And that replicates findings that we have started to see in 2005, that we mapped out the apparent motion trace, and we saw activity along this trace in V1. So that represents kind of an increase of activity of the internal model. Now we're testing with the target stimulus, and that creates an additional activity, shown in this purple line. But if it's at the right timing, then you have a predictive coding, you have an explaining away, shown in the yellow line, that is less activity than just the purple line. And that replicates the predictive coding effect that we have seen in 2010, the Arling paper, and psychophysics that we have tested and so on, showing that the prediction effect transfers to the new hemisphere in time for after the saccade processing. So it's a very quick process, and it should be, or it's good to see that it works this way, because then predictive coding or prediction processing is an efficient, is actually quick enough to matter. And we have now also added a control experiment in which we have a flicker control stimulus, and I'm just wanting to highlight this. So you can see that this is like 6 cycles of apparent motion up and down, then this is the cue for the saccade, but it takes a while till the saccade is executed, and it's timed such, and we select only those trials in which the timing is perfect. So the saccade is finished at this time, and then you have an in-time or an off-time test stimulus and the target. So we do the same thing with the blinking, but now the upper and the lower apparent motion stimulus are shown simultaneously. So you have other internal models, but you don't have an internal model of a smooth up and down movement. And in this case, the target stimulus that is in right or wrong time are not making any difference. They have the same proximity, and so they shouldn't make any difference. And here we have plotted the in-time, the out-of-time minus the in-time responses. So the advantage that you see for the predicted stimulus, or the error signal, if you like, for the out-of-time stimulus is shown here. So in each individual subject, we see this, but in 2. And in the blinking control, we don't see a consistent effect, so there's no prediction effect. Okay, so those are the 2 examples for testing predictions in real time in primary visual cortex for upcoming stimulation. And that covers the question, or that covers one aspect of cortical feedback to primary visual cortex, which is in relation to prediction of upcoming stimuli and in relation to testing its internal models, whether they are fit for purpose, whether they are really representing expected changes in the outside world. However, once evolution has come up with these, let's say, cortex that is able to do this online prediction and negotiating of internal models with outside expected input, it might have used the same or related architecture to also run some offline simulations. And we know relatively little about how they work and what they do and so on, but they need to be probably very closely related and happening in the same areas, because we know there's plenty of evidence that in primary visual cortex, or in early visual cortex for that matter, you have activation in relation to mental imagery, and in relation to dreaming, and in relation, and actually the sleeping brain looks very much similar to the awake brain. So there are questions what these kind of processes are doing and how, what else is happening. So I want to mention very briefly, this would be kind of a talk of itself, a few examples in which we see what else is happening in primary visual cortex as a kind of, if you like, a workspace or a blackboard, which things are projected down and used to manipulate in a counterfactual situation. So the first example is using a version of our occlusion paradigm in which we occlude one part of the visual image. Now we are using the upper right. Usually we use the lower right, but and this is an experiment that combines some features from our lab, but also from Cambridgeshire lab. Williams had shown that if objects are compared and they are shown in the periphery, they somehow, you can read out the information in the fovea of primary visual cortex. So we wanted to understand that. What we have found in studies that I will come to later on is that we can get some information about the scene in the periphery, so in the periphery. So we wanted to combine these designs, and we did this in a very natural way. This is from Matt in my lab. And he designed these stimuli that there are two different scenes. So this is a forest scene, and this is more a beach scene. And then there are objects, a tent or a barbecue set. And the question is, what kind of information is processed in this occluded, what kind of predictions are processed in the occluded areas? And the result, we looked at the fovea part, here shown in red, for scene segmentation. So in the fovea, even though it's not directly feed-forward stimulated, feedback information gives some information about the scene that is processed. So, and we did this in two different tasks. We gave the subjects a task to differentiate between the scenes or to differentiate between the objects. No matter what the subjects are doing and what kind of task they are involved, they always get at the fovea some information about the scene. So this part of the brain of V1 knows whether the context is in a beach or forest, shown here. Now if we go to the periphery, so this part in the periphery, it also always knows whether it's beach or forest, regardless of the task. Now we go to the foveal part again, but we look at object identity. So is there a tent or is there a barbecue set, is the question. And the classifier, using voxels from these occluded region, cannot tell the, okay, we are in the, well, first we're in the periphery. So in the periphery, let's say this is this part, the periphery cannot tell whether there's a tent or a barbecue set, okay, regardless of the task. And the fovea can only, that's the yellow, only tell apart whether it's a tent or a barbecue set if that's part of the task. So that combines, that replicates Williams' findings, showing that if the task is to compare objects that are presented in the periphery, then the fovea will be involved in doing this. So the voxel MVPA analysis that we do is informative of this variation. So therefore, if you like, if you have to do a fine-grained comparison of objects, you're using a blackboard where you project those two images and you're comparing it. or something along these lines. But it's not predictive. You're not predicting a barbecue set in the center of your of the fovea, just while you're doing the task. A study from Lucy, she did a while back, is about face processing. Here we used 20 different faces, and we had either a gender discrimination task or an emotion discrimination task. And we know from, she knows from Philip Shin's lab, that where the informative voxels are around the eye for gender discrimination and emotion, the mouth is very important, and so on. And we used these regions of interest with checkerboards, and we had the idea that there the most information would be. But one of the striking experiments is that in the remaining part, and importantly also including the fovea, it was the best region to decode information about which task the subjects were doing. So were they doing a happy, fearful classification task? And using a few voxels, you could already tell whether the emotion was happy or fearful. And if they did a female-male classification task, it swapped. After you could tell from some voxels already that the kind of content. So again, this speaks to the idea that while you're engaging in a task that is comparing features for emotional expression, face features, and so on, that are certainly not processed at the level of V1, but higher up, but somehow they are projected back into this workspace and make a difference. And most strikingly, of course, this example of motion, there are two of imagery. So, you know, we know mental imagery from series of experiments is involving early visual cortex. And now I can talk about, let's say, VSS meeting. I went to the beach and I saw this purple Cadillac driving by, and in that was a pink giraffe. And you will start imagining something. Maybe you don't follow all the way and had satin glasses on, but you know, it's not predictive in the sense that you would be less surprised if you actually see now a Cadillac driving by, which I described to you. So it's very important that it is counterfactual and does not interfere with your predictive coding. That's also happening there. Another experiment that we did is we asked the subjects to close their eyes while in the scanner and listen to these natural objects. And let's do this in the third. We did a whole series of experiments, and in one way, this naturally creates predictions, if you like, or images. You mentally imagine counterfactual situations. But what we also did is we asked the subjects to do a visual object imagery task, so a cube assembly task, kind of a Koslin task. Okay, let's say I tell you up, up, right, right, down, and then you imagine building blocks, these objects, while you're listening to the sound, and then you'll rotate it by 90 degrees, and then again I say right, right, down, left, left, and ask you whether this is the same object. So you're engaging in a mental imagery task while you're listening to the sounds. And this is one of the control experiments we did. Okay, first of all, the main finding. While we mapped out retinotopic visual areas, V1, V2, V3, and while you do this without any other task, you see kind of a deactivation to the baseline in these areas, which is unspecific to the three different sounds that we presented. If you use multivoxel pattern classifier, you can find information in early visual cortex, especially in area V2 and V3, that is informative for those different auditory scenes. And okay, I'm not showing the control experiment, but we did 5 different control experiments, and one of this was like while you're doing the cube assembly task. And while doing this cube assembly task, indeed, much of the classification in early visual cortex, especially in the fovea, dropped to baseline. However, in the periphery, especially of V2, we could still find significant classification relative to the auditory. So what that means, I think, is that there's potentially 2 systems ongoing. One is if you do this cube assembly task, you're using, again, mental, the fovea to do these high accuracy mental rotation, while in the periphery, you're still processing auditory kind of predictions. You can also think of it if you're at the beach, you hear sounds of the shore, and you are imagining your talk that you will be giving, and then you hear a sound that doesn't fit, you will be alerted to it. So the periphery might just be explaining away everything that is expected and alerting you to those things that aren't expected, while in the fovea you can do mental representations of other tasks. Okay, so that wraps up the kind of additional things that are not totally related to predictive coding. That might be using the same hierarchy to build, to make simulations that are useful, because if you can run some things offline, you then optimize your generative models for predictions further on. You can go through your talks, you can go through your slides, and then you will be better in giving the talk. So that's helpful, right? So that there's prediction for a longer future. Okay, so now let's get back to cortical feedback to V1, and I will zoom in with the different paradigms and look into the layer specificity, where the different signals are coming from. So we have, first of all, lots of areas that are in principle connected back to V1. We know this for 30 years. Most of the connectomes, they are reciprocal, so all these areas to which V1 is feeding to is receiving feedback from those areas. And the layer, the way the cortex is layered, it receives the feed-forward thalamic input in layer 4, predominantly feedback in the superficial layers, in layers 2, 3, and in the deep layers. And it has a certain pattern shown by Markov Kennedy Lab that from neighboring areas, so let's say you have, this would be, let's say we are looking at V2, then the neighboring areas from V1 and V3 would receive feed-forward input shown in red and feedback input shown in blue in layers 2, 3, in the very superficial layers and in the deep layers. But further remote, so if you go to V5 or areas that are not directly connected further away, they will predominantly connect to the most deep layers. So at least that's the kind of pattern that seems to be emerging. From the work of Matthew Larkham, there's one potential neuronal mechanism for integration of this feed-forward signal and feedback signal. These are the very important layer 5 pyramidal cells that have the distal integration zone in the dendrites. The dendrites reach into the very top layers, the layer 1, the superficial layer, where there's huge amounts of dendrites that are coming back from these other higher areas. They can read out this and integrate this and have backpropagating features to the soma. And the more proximal integration integrates the feed forward from layer 4. And they have shown that it has a bursting property if both feedback and feed forward are joined, coming together. And so therefore it's a neuronal mechanism that can integrate these two streams of information. Now the kind of experiments that we have been doing is we do retinotopic mapping, and what you can see with retinotopic mapping with checkerboards is that you will have your activated column next to the non-activated region. And from the lab of Charles Gilbert, you can see that these are V1 cells, and the receptive field are shown in red. But you have also this green surrounding receptive or projection field that integrates the feedback from area V4. And in their example, you have these line neighbors, and if they are co-aligned, they will affect the receptive field of those neurons, even though they're not responding to the features out there. Due to feedback, they will be changed. So you can see this. That the kind of neighborhood relations can be shown to reach very far, much further than the receptive field of V1. And that's the kind of feature that we are interested in looking at. So we want to understand by our design, we are not looking at the feed-forward signal, but we are looking into this region, and we are decoding this region. And by focusing on the non-feed-forward stimulated, we can learn about the features that are back-projected to V1. So that's our strategy. And now we did this with ultra-high field fMRI using 7 Tesla in Minneapolis and in Maastricht. And we do first retinotopic mapping. So this is the region of the occluded space. Then we have the safety boundary in blue that we are not interested because it's a bit, we want to make sure we're really in the occluded area. So this is the bit that is in the occluded area. Then we sample 6 equivocal depth layers of EPI images at a resolution of 0.8 millimeter we are recording. The cortex is about 2 to 4 millimeters thick. So they are not exclusive. They're partially overlapping our 6 layers. But so you could, we use 6 layers just because it's a good compromise. You could use 10 or 4 or so. The fact that we have a 6-layer cortex and that we use 6 layers is not related. Those are just probabilistic depth values, right? It's very important to. Okay, so then we have our voxels that we are interested. I highlighted them here. So those have receptive fields for the feed-forward domain. At the boat, we showed 3 different images: a boat, a car, and a market scene. And so there's rich information. There's a steering wheel, there's the front of the boat, or there would be a person walking by. In the occluded condition, we have the white screen and no information. The question is there feedback coming from higher areas? We have shown this in the 2010 paper that there is, but now we are wondering in which layer is it talking to. So those are 4 subjects, and you can see in red it's almost ceiling because all of these layers of the layers are shown from purple to red. So purple being the most deep layer, and red are the superficial layers. And you can see that for feed-forward, you have rich information in all layers, but for the feedback regime, it's only in the outer layers. So the superficial layers receive this feedback. Those are the average. It's important. There's a big question if you do layer-specific high-resolution fMRI. There's a big debate whether the bold signal, as such, that you're working with, has a different amplitude in the different layers. And gradient echo images are known to have the certain feature that if you're in the outer layers, you have a stronger bold response. You have bigger vessels shown here, and gradient echo sequences are sensitive to this in creating a stronger bold response. So we can replicate this in our feed-forward regime. So when these voxels that we're analyzing are looking at steering wheel and so on, you see that the amplitude is higher in the outer layers. However, the MVPA information plateaus. So it's not the same pattern. It's not increasing. So amplitude does not say more information. And in our occluded paradigm, we don't have an amplitude. We are just on baseline. Yet we have a different profile. The profile of information, MVP information, increases. So those 2 values, amplitude and information, are unrelated. Moreover, we have used another sequence, Gray's 3D sequence, which doesn't have the property to give higher amplitudes in the outer layers. And doing this, it doesn't have so much contrast to noise. It's more difficult. It only worked in 2 out of our 4 subjects. That even the feed-forward, you see the feed-forward classification drops now dramatically. It was ceiling before, now it's at 50%. So that's the sensitivity of the technique is just not as good. But for those subjects that feed-forward works, feedback has also the property to only be in the superficial layers. So it replicates, in essence, this finding. Okay, so what else we did was we measure the precision of the classifier in a parametric design. So first we have, let's say, these 2 stimuli, a car and a kid, and we have the classifier trained on differentiating these without shifts, and then we start to shift the image for 3, 4, 6, and so on degrees, and run the classifier again between car and kid or people in the shifted version. So first we train the classifier in unshifted, and then we test it in the shifted version. So at some point it needs to go down to baseline. But by this parametric design we can see what's the intrinsic precision of these feedback signals. And using 3 Tesla we came to the conclusion that it's there's a step when you go from 0 to anything, to any shifts. So there's some high information, some high resolution information, but there's also a drop off after 4 degrees where it becomes baseline. So therefore, we think that these kind of predictive signals are around with a precision of 4 degrees. Now we replicated this also in the 7T environment, and you can see the pink line is the only line that has small shifts, 0 to 2 degrees, whereas the blue and the green lines are for higher shifts. And only the pink can classify, and again, only in the outer layers. And now we have upscaled. That's Tyler's project. So before that was Lucy's project. Both of them are here, and they are presenting posters. And I think they will be up again today over lunch, if I'm correct. So you have a chance to speak to them. With Tyler, we have upscaled the used images. Before we have used a very small sample, 3 images and so on. We use many repetitions, so these experiments take quite a long time, and we're now upscaling it to hundreds of images, and this will eventually then be also available in the database of the Human Brain Project for modeling and so on at high resolution at 7T. But this is now a 3T project with 24 different images of 6 different categories in 2 flavors, one more near, one more far. We used categories and we used near and far because it has already been shown that these features can be classified in V1. So now our question was, can that be classified in the feedback signal, in the occlusion? How much information is in the occlusion? Do we have individual image information? How rich is it? What can we learn about the feedback? And I'll summarize these data in a very elegant way that Tyler came up after speaking to Jim, actually. So first of all, you see these 6 different categories, near and far. And when we do classifier for categories and near and far and individual images, all of these works. But near far is the most fragile one. If you do extrapolating from your image set that you across images that weren't in the training set, it will fail. The category will still work, so you can train on 3 beaches and then on 3 beaches, 3 buildings, 3 forests, and then test on a new beach and a new forest, and it will work in the occluded paradigm. But there are many other things you can learn about these features. It's now in bioRxiv, this paper, so you can read up more of the details. Here's a representation of similarity analysis way of showing the results, and V2 and V1, the similarities. So here you have beaches, buildings, forests. The forest is always the nicest to see. This is for feed-forward information on the left. This is for feedback information. So feed-forward is very similar. V1, V2 are pretty much doing the same thing when they are stimulated, and forests pretty much always look alike. So the confusion matrix, they're all very much alike. In the feedback signal, you also have this. So this is now in the occluded, and the forests also in the prediction look a little bit alike. So this may be a sanity check. You see along the diagonal that it's possible to individually classify these different images. And we compare this to computational models. And so there's the Y-ball model, there's the GIST model, HMAX model at the layer C2. And we feed in our stimuli, and these are the representational similarity analysis for the feed-forward stimuli or for the occluded section of the image. But this, Jim said, well, that's not a very illustrative way of showing the dissimilarity matrices comparison. So therefore, Tyler projected this into a multidimensional space, these dissimilarity matrices, and now each point is a point in the high dimensional space, and back projected into 2D space along the two principal components that are extracted. So now you see very interesting features by this projection, that the models are all looking very much alike. So maybe in this version it's better. So they're occupying a space that are greatly overlapping. So that's the way we think V1 works. And the actual data for the non-occluded, so the feed-forward information, is shown here for V1 and V2, and it's greatly overlapping. So V1 and V2, if fed with visual stimuli, are doing pretty much the same thing. We have seen that in the confusion matrix. That looks pretty much alike. But very interesting, this is not overlapping with the models. That's also something Niku showed. Models are a little bit off from what's actually happening. So that's interesting. So the challenge will be to move these models to incorporate these regions. But then what's also interesting is that the occluded areas, V1 and V2, first do two different things. V1 and V2 are not doing the same thing. And we have found in all of our experiments that the classifier often worked better in V1 than V2, and there are differences in V1 and V2, but only in the non-occluded, so in the feedback regime. And we have no clue what they are doing, right? So that's projection for future research is to develop models. It's not reinstating the feed forward, then it would be overlaying here, and it's not doing what the models are, describing it should be doing. So if the models would describe the features that are missing, and the occluded would just really fit in the missing pieces, so to say, then these patches would be over here. And with this, I'm just wrapping up. So I've shown various experiments, kind of a proof of principle, that internal models generate predictions in the cortical entry state for visual processing, area V1. It shows, in the case of apparent motion, in moving scenes, with auditory stimulation. And much of this is task dependent. So if objects and scenes are manipulated and you have to think about whether this is a barbecue set or a tent, then this is projected to the fovea. You're processing faces. It depends on the task, whether you're a gender or emotional discrimination task, that will matter to V1. So that must be a feedback feature. We know something about the mechanisms. Feedback may amplify or disamplify the predicted information. It's happening in the superficial layers of V1. The precision is somewhere around 4 degrees of the feedback signal, whereas the feed-forward signal has a precision of around 1 degree. It's fast enough to sustain eye movements. It can be destroyed by TMS to V5, the apparent motion paradigm, which is something I haven't shown you today due to time. It's in the auditory experiment, we have some evidence that it's categorical, also something I haven't shown. With the occlusion paradigm, Tyler's experiment showed it is categorical, so we can extract, there's something about beaches and offices that is typical also in the feedback projections. And the models are yet to capture the complexity of the feedback signals. We are thinking of some applications in the clinical sense. If you learn about the feedback signal and of the predictions, it might be very useful in case for schizophrenia, aging, autism, all of those cases. There are hypotheses that the predictive generative models are affected. There's some interesting literature. We have tested one experiment with schizophrenia. We didn't see a difference. Maybe it was too basic, but that's certainly something that the field will explore more. And with this, I thank my lab. You can see the different contributions. And as I said, Tyler and Lucy are here. So if you have further questions, they're happy to talk to you. And I take questions, of course.
We have time for a few questions. So, comments?
Really exciting talk. And so we have this local dynamics in V1, right, which already serves as prediction model. So it's probably dynamic system, which it kind of just adapts to our dynamic stimuli. Then we have some probably high-level inputs there which modulate it, right? And let's say with this really cute experiment with auditory influence back, is there any mediation? Because we have V1, probably it has its own dynamic kind of stable state, how it works. Then we have V2. How much do they talk to each other to share this dynamical information? Because all those superficial layers, they're more local, right, than any distant connection. Auditory, does it go directly kind of influence? And did you look at the, let's say, connectivity, some functional connectivity of those?
So starting from the last question, we have only some indication that it's happening more on the periphery for auditory, which matches to the known anatomy in monkeys to be, there are some direct links. We are now also running high resolution 7T with the auditory paradigm. I showed some preliminary result at HBM, but it's as yet a bit inconclusive, very pretty. A few subjects show, that you have more connections to the deep layers, but it's too early to be very conclusive. We are also exploring different techniques to see where the signal is coming from. And in the case of the tent versus barbecue set study, Matt did actually a very nice follow-up experiment, or not experiment, analysis, in which he looked at the kind of information that's in the occluded space and compared that with a searchlight to the other visual areas that we mapped to see where is the same kind of representational similarity matrix than in the occluded part, and found LO to be the closest match. So that's a very good way of showing that actually the information for this kind of task was including LO, or looked like an LO signal, so to say. That was the later part of your question, and the first one was...
it was probably a statement or something.
Right, how they all interact. I mean, I think one thing where I only have intuition and so far I haven't really... I mean, one of the dark matter, or what's missing in all of this is memory. Somehow we know memory needs to be also in cortex, right? It's not only hippocampus, so it plays to the whole cortex, and since cortex looks alike wherever you go, so it should include V1, it should include all other areas. And what does that mean? How does it play out? How do you, if you do mental, imagery, you go in through your memory and so on, and create these kind of situations. And so that's probably what's happening, and all the different areas are contributing different aspects to that. And these kind of memory and feed-forward models, because when you do a feed-forward model, you will need your memory, so you generate out of memory the predictions. So these kind of negotiations is probably happening somehow in a system that can be switched on and off. So that's the kind of, I think in those layers, we're always talking about perception and the kind of online mode in which it compares the input, and that makes a very dry reality. But I think that even rats have this, that before they go in the maze, they mentally play out left or right. So the place cells kind of reward. Okay, let's go there. No reward, apparently. So that's an efficient way of playing it out mentally, and so therefore I think that's what's happening there.
Or maybe we have a caching mechanism, like different levels of caching, like we have with computers, right? We figured out that we can perform computation faster if we keep whatever we need for present task, right, close by, and this dynamic system could encode. That's why I wondered, let's say in first trials, did you see any adaptation effects through the trials, right, with all these effects? Because it could be also that we recall the memory from, there is a beach, right? But next time beach comes in, right, we have already this dynamic system adapted for, beach again, and this one, so.
Yeah, we looked at this in some way, whether over the trials the predictions become better, and we also had one set of subjects that hadn't seen the full image and only the occluded one, so it couldn't be from memory that they're kind of filling in what they had seen in other runs. But that didn't make a difference. Since we had more conditions in those subjects because we didn't have the feed forward, all the classifier performance was just much better. Reason is we just have, this is knowledge we have acquired, right? I mean, you see the tennis player, you know so much, and that's what's already in there. That's why we use natural images. If you use, maybe more abstract things that you learn later in life, like numbers and letters and so on, maybe that's more a low, maybe a later stage. But these natural images, well, okay, we need to test. I can't say.
Okay. Thank you very much.
Yeah.
Okay, so is there any? I think we have just one short question, so go ahead.
I don't think it's going to be short. But anyway, I'll try to keep it as short as possible.
Answer that.
I think we have.
Thanks for the talk, Lars. One thing I was wondering is whether so you've shown very nicely that you can decode information using the top-down stream and using imagery. It'd be nice to now try to understand mechanistically how is that information sort of getting there in the first place. And I was wondering if one might be able to use deep neural nets actually for this. And so I just had a wild idea about maybe training the classifier based on the feed-forward inputs, and then using the highest level that corresponds to whatever image is held in working memory, and then just shifting the weights around so that you back-propagate activity to V1. Perhaps also, as Jim was saying yesterday, using constraints on the known anatomy. So we don't expect the feed-forward weights to be exactly the same. So we might apply some kind of kernel to make them look more like what we know from anatomy, and whether or not then you could actually predict activity in V1 or early visual cortex, or perhaps some other way of using the deep neural networks to gain a mechanistic understanding, not just what information can be read out, but sort of how is that being built up as you go down the hierarchy. So sort of reversing the flow that we typically use with those models.
And you mean electrophysiology data to compare it with?
That would probably be quite informative data, but I'm sure you can. Well, I don't know. It's an open question whether it can be done with fMRI.
Yeah. I mean, that's what we're trying to do now with a kind of encoding model that we create. We use, first of all, much more stimuli, so that we have like 300 different stimuli. Then we classify the different boxes, how they would respond to feed-forward stimuli. And then in the occluded paradigm, see what they are responding to, kind of reproduce or the kind of filled in information. We kind of project back what they would usually code in the feed-forward sense. What we can't do is that dynamically. So we sacrifice some ways with each method. So the kind of temporal integration, how that pulls out over time, we don't really know. We don't have a very good grab of this. But yeah, using neural nets to simulate would be wonderful.